successfully with lot of hurdles. Not only that, he proceeded further to Scotland to get advanced training in medical science, came back and served for India. This should be the dedications. So we should have the patience. My dear students, particularly the young, you should have the patience getting here, and you will be the future of this country. Let's start from this auditorium, from this Utkal University. I think without taking much time, we must invite our esteemed speakers to deliver on the different aspects, and let us learn from them. Thank you, everyone. Vande Utkal Jana.
Thank you very much, sir, for sharing the valuable insights and inspiring the young minds present in the audience with your address. Now, before we begin the distinguished lecture session, we would like to felicitate our esteemed guest as a token of our gratitude and respect. I request the dignitaries on the dais to kindly come forward for the felicitation ceremony. Sir and madam, please step ahead and join us for the felicitation ceremony. I request the Honorable Vice Chancellor, Sir, Professor Jagneshwar Dhanpad, Sir, to kindly felicitate Professor Sameer Kumar Patro by presenting a Uttariya and a memento as a gesture for the respect and a gratitude from the Utkal University. I request Professor Dhanpad sir to kindly present a memento to Professor Bedang Das Mohanty sir as a token of our gratitude and respect for accepting the invitation today. I request the Vice Chancellor sir to kindly confer the memento to Dr. Sushil Kumar Singh sir for gracing today's event and accepting our invitation. I request sir to kindly do the honor of felicitation to Dr. Priyadashni Mishra Madam for gracing the occasion. May I now request Professor Sameer Kumar Patra sir to kindly felicitate our Vice Chancellor sir by presenting a memento. Our sir is too modest. He's refusing to take the memento, but this celebration was sir's brainchild. Thank you very much, sir. I request Professor Bedang Das Mohanty, sir. I request Professor in felicitation of our Revit Chairperson Post-Graduation Council, Professor Mitali Chinara Madam, our Director R&D, Professor Joy Dev Dinda, sir. Thank you very much to all the dignitaries on the dais. Now with this, we have come to the most awaited session of today's event. And it is my proud privilege to invite our first distinguished speaker for today, Professor Sameer Patra, sir. Sir, I request you to kindly address the gathering with your distinguished speech, sir. And I also request the dignitaries on the dais to kindly join the august gathering for the better view of the presentation. Thank you, sir.
Good morning, one and all. It's a proud privilege for me to be here at the famous Utkal University. And special thanks to Professor Dandapat, who is the Vice Chancellor of this prestigious university. I also thank all the dignitaries on and off the dais for an auspicious gathering here to celebrate Women in Science, the Viksit Bharat 2047. Before going to the main course of the lecture, I would like to see why Viksit Bharat and Women in Science. You know, if we look into the history of India, we will find that India was the premier place of education, science, and society. Better this side. If we look into the history, you will see Nalanda University, you know. I will not tell, I will just name that one, you all know. If you go to the civilization, you know, in the civilization, and so on. And if we go for the Ramayana and Mahabharata, when we will go, or even the other way, why women in science, particularly if we look Goddess Durga, when all the gods were defeated, they surrendered. Mom Durga, you please save us. So, you know, the powerful, women are always powerful. If you neglect them, you are the loser. And the current day, our government, precisely Modi ji, has entrusted and he has facilitated the honor as well as the utilizations of the power, women's power. So with this, I would like to say about that precisely National Science Day is celebrated in India in February 28 to mark the discovery of the Raman effect that was discovered in 28 February 1928. And this is Sir C.V. Raman, the picture. Just to give him a preview, the principle of Raman effect, as you can look, in those days when science was developing, you know, in 16th, early 17th, then 18th, 19th, 19th, and 20th century, when he was looking, he knew that certain effect is going on when light passes through the matter, what we call the light scattering effect. And at that point, the famous Rayleigh scattering was done. The scientist he did it. But Raman was such a genius, he don't wanted to stop there. He was not stopping there. He wanted to use that what is happening thereafter. So he used other materials and he found out that there is certain scattering. The absorption of light, you know, nowadays we know it is wave pattern and there are two hubs of the waves. If it is differentially affected, the light trajectory becomes deflected. And due to this reason, many events happen. But here what happens is that I am a diabetic person. So when I talk much, try it. That's why I need some water. So he was trying to figure out all these things, why it is deflected. Nowadays we know the applications or materials characterization, and precisely these are happening due to vibrational state of the atoms and molecules. This is the true scientific sense. So nature, nurture, and science. We know universe. Heard about it, but we can't imagine. Whatever we know, that is little less. Universe is so vast that we know it. We know whatever when we go through books and when we listen to a learned person, we think that we know many things, but really we don't know anything about the universe. So the nurture in the sense in the earth world, as we discussed that the way the baby nurturing, etc. But we are unable to nurture the nature. Actually, the universal concept, when the concept of the universe coming, we are unable to nurture anything. And in science, precisely, will come when we want to explore anything, a philosophy should be there. That means you have the power of thinking something, and when philosophy comes, a thoughtful vision is, we mean that one. It leads to an hypothesis, and when hypothesis is proven, we call it's a fact repeatedly. And now in science, in our limitations, what we call vision with materialistic reproducibility. I will show you this is a total failure when we will think about the universe. And it becomes a fact, and that is a must in our conceptual thoughts. Now, what is universe? The universe, if we look here, a James Webb Space Telescope that has been published in July 25, you will see what is a lot of dark space, light, and bright sites are there. What are these? I will come later on. Just you see another telescopic view. Now, nowadays, in those days, in the say 200 years back, when scientists were thinking about that with different kinds of telescope, they just spotted and drawn it. Now we have the data, we can calculate it in different learning modes by computational adding and so on. So we can draw better spins and quantum wise the vision of the universe. So here, when we look, the universe is mostly composed of different types of galaxies, largely a space, and there are light and dark materials. So here spiral, Barrett spiral, irregular, lenticular, then peculiar. When you are undefined, you are not able to define, the scientist, the astronomer name it a peculiar shape, and here elliptical shape. So here I will give you an example. Although there are, we are unable to detect, but yet we can do something. Here we can see the galaxies are in different, we are in a galaxy, we know that is a Milky Way galaxy, where the solar system exists, and there are various stars and other issues are there, but here galaxies can interact. That is the most nicest thing about that. But what we are observing now, this one, it happened maybe say billion years ago. So because of the what we perceive, our perception is that due to light. So when light comes, it takes several light years. Say that's why what we are observing now is not now. It's a lot of billions of years ago. So that is the explanation of that. Now here, if we look, globular clusters in the hub that is beyond the galactic core. When we observe, we differently name them. Here I took it from Animalia Life Club, Milky Way Galaxy. The new general catalog nebula and clusters. What is nebula? Nebula is the initial phase of star formation. So it is composed of hydrogen, helium, and various others, and little bit energy. So different nebula and stars are named here. As you can, there is a PAL also. PAL means phase alternating line. Then there are also ESOs and VLT. These are European Southern Observatory Laboratory Large Telescope. It was deciphered, and so on. So what is the composition? And from the beginning of our understanding about the Earth, and then when we tried to think about, we matlab here, they say 1,000 years back when scientists tried to it, that one. So what is how the universe is composed of? The universe was no longer, it was never smooth, uniform, and so on. So it took different types of shapes due to the energy dissipation in different projections. So in that way, you will see like a volcano when it coming in the little form, you can imagine that in the universe, it was starting, in the starting days, it was like that one. So here compositionally, if we look into this, there are dark matter about 27%, then dark energy 68%, and here ordinary matter is about 4%, hydrogen and helium, 5% ordinary matter, about it helium and hydrogen and various other gases, less than 1% are stars and 1% are other things. So this is the overall composition. So now we will go the Big Bang expansion little bit. I will just give a glimpses of this one. So if according to the quantum calculations, quantum fluctuations here, if it started at that time after a light pattern at 13.8 billion years ago, a Big Bang expansion had been done. The first stars forms about 400 million years ago, and development of galaxies, planets, etc., so and so on. So if you go to the Stephen Hawking's concept, it started from a point that is hard to believe. But at certain point, you have to believe where it was started. You can say that it was existing, but in certain way, you can say that no, it started from a point. The point, although when we try to equalize it with the mass, that will not give in our concept, it is not possible. The first thing from a point, and precisely when there was, I mean, nothing, how it is giving us. So in certain ways, I am compelled to believe it, and in certain ways, I don't believe it. So this is the scenario, but it is scientifically, it is very hard to prove, because as we have realized that it takes, light comes, light is the only detection in case of our scientific world. So when it comes from the other object, it took so many years that we cannot say. So, but yet we are getting something, the concept, we are endless thought and is coming in that way. So we are getting it. So now I already have given the whatever I felt. Now the cosmic background explorer and various other points where the large telescopes are placed there and invented such a diagram. There, here is cosmic microwave, then Wilkinson microwave, and various others are there. In Keck School in Japan. So difficulties, yet resolving the terms of technology is that one. There are difficulties. I will skip this one for explanatory. If you look into this one, Joanna Thompson published in February 7, 26. A team of February 7, 26, a team of researchers using James Webb Space Telescope, they found it that invisible scaffolds are there. The scaffolds we know we can make in the universe, the scaffolds, whatever thing in our earthly space, in our laboratory, it is used. The thing, so universe is composed in such a way that it is happening. Now coming down, after the Big Bang, it was so large heat, lot of matters, but most of the matters are dark matter, dark energy, and little portion is our matter, what we are facing day to day in our Earth's surface. So it was cooling, and cooling. Hydrogen and helium was the most important elements, and then elements were also evolved in different ways. So here is our Earth in the third one from the sun trajectory. Now coming to when the Earth sufficiently cool, we found the elements. It came in our chemistry world, the elemental world. So quickly you can see the timeline. I am passing quickly. All of you know some of the great scientists I mentioned here. Then you can also, those who have interest, you can go to the magics of elemental discovery. The funny thing is that here, if you look, many of the elements like copper, lead, etc., those who are abundant in surface, we will hardly find who discovered it. It was a social discovery. According to your need, the people are trying to use, and it was named in that way. And there are various others. Quickly passing because of the time limit, I thought it will be 45 minutes, but I learned directly from the Vice Chancellor, sir, that I have to complete it in 35 minutes. So, origin of life, that is most important. I'm a biologist. I'm a chemist turned to a biochemist. Then we are working in different parts of disease biology, precisely genetics and epigenetics. So, origin of life is fascinating aspect. And you see, the first composed theory was given by Alexander Oparin and J.V.S. Holden, the primordial soup theory. When we know the elements and by lightning and others, the elemental composition is giving molecules, and then molecules is giving large molecules, then macromolecules, and so on. From the analysis of living organisms, we all know that one. And the most important thing, what they suggested is that early oceans from simple organic compounds formed through chemical reactions among gases like methane, ammonia, hydrogen, and energized by lightning or ultraviolet light exposure. And in 1953, Urey and Miller experimental supported that one. We got in our Earth's surface. The thing is believing until and unless we are able to prove it, we are in doubt whether it was there or not. So Urey and Miller give a strong foundation of J.V.S. Holden and Oparin model. there is a special attention, if the, it will be a serious omission if I don't mention here. J.B.S. Holden, a scientist from UK, he worked here in the, he was son of the soil, you can tell. He worked here in Bengal and Orissa. And until his last breath, he was in Bhubaneswar. And he was died in 1961. So, the whatever we are seeing, operant model and J.B.S. Holden, the primordial soup theory was proposed from India by his work and knowledge. At that time, he was a superannuated scientist from UK and he migrated here. There are a lot of political and other hazards. I have no sufficient time to discuss about that. I studied a lot about these scientists. Then it is coming the deep sea vent hypothesis. The same thing that it was under the deep sea level, a lot of chemicals, organic compounds were there, and they somehow mix up, and in this way it was started. And another was is that to me it is not a funny thing, but it's really skepticism will come that panspermia, that means the other satellites or other galaxies, the life started first, then somehow it come. But somehow means how, that is pretty difficult to understand, because how the time it takes from light to come here with such a speed, say 3.8 years and so on. So it is pretty difficult to say that panspermia has originated the life on Earth's surface. But yet some philosophers have put forward into it. Then will come the clay hypothesis, that same you think that various other, then iron sulfur, the electrical spark hypothesis. It has some connections with the previous experiments has been done by Miller and Urey. Then RNA world, as you know, those who are biologists working here, that RNA has a little bit enzymatic activities. That's why RNA enzymes or RNA enzymes, that's why ribozymes. That's why the scientists who are working about that RNA was first created. But RNA cannot be, RNA is composed of a lot of nucleotides and so on. The nucleotide first be forming. It again comes back to the original hypothesis given by Holden and Oparin. Now, lipid world hypothesis, it is a little bit not confusing, because cell membrane is there, and without membrane cell do not exist, and cell membrane is lipid bilayer. You can look into this one. Then coming thermodynamic dissipation theory, you have to have belief on it, because whatever is coming is the energy source is there, and precisely second law of thermodynamics dictates many things about the life. Then coming radioactive beach hypothesis, we cannot exclude it, something is there, but as a whole, life originated from all these theories mixtures, no one can be excluded. Now coming chemistry, elemental world, the molecular world already we have gotten, then macromolecular world, then crowding and mixing, as it looks. So coming to the materialistic world, when we look or we analyze the living organism, we find carbon, hydrogen. You look into the level 1 elements of lives. Here this part, carbon, hydrogen, sulfur, nitrogen, phosphorus, oxygen, and several other, but these are the main composition. Then we will come to the monomeric units. You will find nucleotides, amino acids, cholesterol, sugars, and then various enzymes. Then you are coming macromolecules, DNA, protein, you are getting lipid and cellulose, and then coming the level of supramolecular assemblages like your chromosome, the plasma membrane, the cell wall, and as a whole you are getting the cell. All of you are associated with that, those who are doing biology. Then again, you can redefine your concept of origin of life. The 4 main stages of early Earth that made life possible, that is abiotic synthesis of small organic molecules, then coming, the synthesis of polymers from the monomers, the formation of protobionts. Protobionts, that is complex of different kinds of molecules, what we call carbohydrate, protein, nucleic acids, and so on. And the beginning of self-replicating molecules, that is self-replicating, DNA, etc. But this needs enzymes and so on. So this is a little bit handwork of mine. You see hydrogen, water, CH4, NH3, and so on, glucose, DNA, RNA, all these. And if you mixture in a closed flux and then electrical spark is given, you will find different kinds of molecules. That is for sure. So origin of life, formation of lipid bilayer. When we look into the living cells from unicellular to multicellular. Cells are the unit of life. If it is multicellular, there are different types of cells, but all cells have a barrier or boundary, that is lipid bilayer. So how it was formed? If we think about the early life composition, now the characteristic is that is amphiphils. It has one surface, one head that is water loving, and the other surface like the two tails, like my two fingers here as I am showing. These are the tails. We call these are the hydrophobic. So when you put any lipid molecule in water, generally water loving phase comes down and we find a monolayer surface. The water loving part, the head part we call, that is in the water surface, and the two legs are protrude upward to the surface. And it has in a motion fluctuation, but yet still rigid. That is hydrophobic energy is stabilizing it. Now if you think that this layer was there in here, if you see the tides in the water surface, just if we go to the Puri seaside, we will see a lot of tides are coming. Just think about that in the barrier where soil is there, one surface like barrier is there, and there was the monolayer. Then a tide comes, another monolayer faces into that. You are exactly giving you the lipid bilayer. So I love this part. Now life is what? We will define. What is life? Life is water, pH, buffer, and biomolecules in our concept. When we come to the laboratory, when we analyze any living person's living body. So what is water? Any student? H2O? H2O? No. H2 is the unit of water. If you look into these H2O molecules, when they are conjoined, a cluster of H2O is water. They are clustered through hydrogen bonding. That is water. What is pH? Negative logarithm of hydrogen ion concentrations are no. pH is a scale which tells us the intensity, that is of acidity or alkalinity of a solution. Yes or no? And this scale is made of pH is equal to log of negative logarithm of hydrogen ion concentration to the base 10. So that is the issue here. Now, what is the value of Kw? The scale when you define 1 to 14, why it is that? We know that Kw, ion product of water, do not exceed 14. That's why the scale is up to that point. So I think I have given you the concept. I don't need more about it. Now what is life? And in a living organism, precisely from bacteria, E. coli through human being, you will find all these characteristics. That is a program. Program is there in your DNA and RNA. Every living organism program. Then you will find the improvisation is there always. Then you will find compartmentalization. Energy is essential. Regeneration capability should be there. Then adaptability should be there in any kind of adverse environment. And there is always a stop, seclusion we call. The biochemical reactions happening within same and enzymatic reaction and so on, all are facilitated by that. So these are known as 7 pillars of life and proposed by Daniel E. Koslan, a renowned enzymologist. You know Koslan induced fit hypothesis of enzyme. So life evolved after the Big Bang. Quickly I will pass. Evolution is coming and the human being through these spaces. So again, in a human being you know, but I was always surprised by this development of a beautiful butterfly. You see, it goes egg to caterpillar to crystal, and then butterfly is coming. So always there is certain changes, the regeneration, evolution, all the characters feature what I have told in the definition of the life, it is coming in the form of this one. Then if you go to the cladistic, that means tries to show evolutionary relationship based on that. I learned from my student days, one scientist, Professor Shamapada Sen of the botany department, he told if your research on living organism or any kind of biological aspects cannot correlate with the evolutionary pattern, that research is incomplete. So here if we look carefully, the evolutionary relationship based on the physical traits, Here you will find all these things. Just if you look at the tail, head or legs patterns, you will find different kinds of characteristics. Then here similarly the plant characteristics you can tell that is known as cladogram, the developmental aspects here as you can see. Then coming the brain part, what we analyze precisely, most important issue, we have frontal part, temporal, parietal and occipital part. So here I see that madam is there in medical science. If I am wrong at any point, you please tell me later on. So here it is. So what is that? Here most nicest thing is that if you look into the development of the brain pattern of the organism from different evolutionary perspective, you will get a lot of information that what evolution means. So here this is Paleozoic era, the down one, the first of the 3 geological eras of Phanerozoic eon lasting from 538 to 251 million years ago. Then will come the Mesozoic era, the middle one, then Cenozoic era. We belong to the Cenozoic era. Lot of characteristics if you find from these two, Paleozoic and Mesozoic era, then you can figure out how it is evolved to the Cenozoic era. So evolution, change of species over time, change in the heritable characteristics of a population over succeeding generation, and you have to remember an organized plan. In early life, nucleic acids like DNA that describes both ingredients and the kinetics of interaction needed for the system to persist over time. Then unicellular and multicellular organisms, if we look, we come to the shape and size. So I am quickly passing, just you can see a glimpses of that one from atom to human being, organized in different ways. The different dimensions are there as per the relative scale. Now, I am a cancer biologist. Precisely, I started cancer research in UCSF, University of California, San Francisco. And that project was on epigenetic characterization of prostate cancer. So, the prostate cancer cell, if you look carefully, here the 10.3 micrometer is also significantly smaller than the size of a culture cell, that is 18 to 21 micrometer in diameter. Now, what is a gene? My dear students, anyone? Sir, hereditary character. That is, I realize that hereditary character. But in chemical world, what is the meaning of that one? That we know, sir, DNA or RNA is also there, that is gene. But DNA, a segment of DNA that encodes a product, that is diffusible product, that is known as gene. So it could be RNA, and that RNA can be further translated into protein. But not all RNAs necessarily needs to be transferred. We know tRNA, we know various other mRNA and others. Nowadays we know all these things happening. So a segment of nucleic acid, mostly DNA, which encodes a diffusible product. Now, this is a cartoon, I don't have sufficient time. So human genome, if you look at the karyotypes, 46 chromosomes are there, 23 pairs. So among these, up to 22 are known as autosomes, and rest one is XY pattern, you know all these things. It's a nucleic acid wise, if you know the nucleotides, bases, sugars, you see the nucleotides here, just look into this part. It's sometimes coming, sometimes not. The green one in the right down panel. So here OH group is there in the down. If you remove one H, that is deoxyribonucleotide. So this is the nucleotides. Now the DNA itself within the cell, it is so long, if it is not folded, it will not be confined in the cells. So DNA is composed in a confined cell, what you call chromosomes in the G1 state. But this is a fold of your DNA double helix at 2 nanometer in diameter, then it folds within 8 histone proteins, 4 types to each molecule, histone octamer. It gives, then it defines in a different way. The nucleosomes coil to form chromatin fiber, 30 nanometer, then 30 nanometer gives 300 and so on. Then ultimately it is wrapped into condensed chromatin space, as has been shown here. DNA 2 nanometer, core histone tail, all these things, patterns are coming. So how it happens? If you look into the histone proteins, they are more enriched with lysine and arginine. They have the positive side chains. And if you look into the DNA, the backbone, sugar phosphate backbone, phosphates are largely negative charge and these are positively charged. So they wrapped ultimately with the force positive negative interactions and wrapping of the DNA, nucleosome forms. So what is epigenetics? Epi, the terms words beyond something, side or over. Epigenetics in the sense, in the large sense, any kind of phenomena beyond genetics or the involvement of genes is epigenetics. So epigenetics in the sense, but the molecular sense, it is a layer of information by DNA methylation or in the chromosomal chromatin modification pattern. So it was initially proposed by Conrad Hall Waddington. At that time, DNA double helix was not proposed in 1942, and he was a developmental biologist. When he compared the expression of the genes with the rolling of a stone from the uphill, and if you see the different valleys are there. Once the stone is in one valley, it cannot itself come again back until and unless it is put back there by hand. So similarly, from zygote to different kinds of tissues and others, once it is formed, it cannot be going back to the zygote. So that was the concept. So DNA methylation, as I already told, a layer of information. Precisely, there are a lot of, on all the 4 types of bases, they can be modified at different sites by chemical modification, methylation, acetylation, and so on. But here, what I am talking about is largely the cytosine 5th carbon position, the red part, the trimethylation. Only originally it was, and you can this methylation gives in the CG sequences the promoter enriched region. When methylation is there, specific types of protein binds known as methyl domain containing binding protein. And as a result, it blocks the entry of the transcription factor and RNA polymerase. As a result, when DNA methylation is there in a large crowd, the transcription stops. That is the mechanism of silencing of gene. And here if you look, the histone modification, methylation, acetylation, ubiquitation, so many patterns are there. In the corner of the second panel, you see methylation, etc. has been given. And it is, DNA methylation is simple and easy to understand, but your histone methylation, which one will take over the which stage, it is not common. It is a gene specific wise and signal specificity wise, it takes the tracks and changes itself accordingly. So in largely when we talk about the genetics, it is the inheritance of genetic pattern from one species, the ancestor from the other, or the evolution from one species to other. That is genetics. And when we will come to the epigenetics, largely it dictates or tells us about the expression pattern of genes which gives the phenotypic characters of in every individual organism. So here after fertilization, you will see the zygote is formed. So from the germ layer zygote, the zygotic genome activation occurs and various germ layers is giving us in different ways 3 main ectoderm, mesoderm and endoderm germ layers form. And from that we give different types of tissues and overall organism then develop from these 3 germ layers. Here as you see, the first slide was a galaxy, the second was this one. I find why I was interested. Now the common feature is that I'm a biochemist, I'm doing epigenetic research, cancer research and so on. Why I took that one? It is not a one day or 2 day. I am really hunted with that, the space and research. And I find lot of mystery. What I find in the universe, I found during the developmental phase. You see, from a single zygote, how you are getting a gigantic organism, elephant, human, and whatever is there. And tissue specificity is so far, so nicely, and it is tuned in that way. So initial stages, whatever you know about the cell cycle, cell division, it is not exactly what is happening in the somatic cells. So I think I can get more 5 minutes more. What is the time now? I took more than 35? when you are talking about science, precisely when you are in your own field, it's pretty difficult for old guys like us to stop. But yet I have to stop somewhere. But I'm giving you some notions about this. So if you look into this zygote, it's a fantastic scenario I could find. And what is the more mystery is there, either in the universe, our understanding, or here in from zygote to a living organism. Mostly, I told you I work on epigenetics, and epigenetics, it dictates the gene expression pattern. These are the clusters. It was done, I've done in my lab also. These PN means pronucleus. PN means pronucleus, the maternal and paternal. Now, when just after fertilization, what happens is that your paternal genome, you look at the paternal is the green one, it drastically demethylated, whereas maternal genome remains unaltered, no change precisely. Then they mix up and it forms the chromosomal pattern. So that is the thing. The always, if you look into the just out of the context, just a joke. If you look into the family, all the environmental burdens and thirst are coming to the males, not to the females. So here also, it is from the beginning. You see, the paternal genome is drastically demethylated, whereas maternal part remains unturned, untouched, or there is less dynamics on it. So here is, I am skipping all these quickly. So now, what is the biasness? Your glycolysis is there. It's a regular pattern, that is housekeeping pattern, and another is specialized. If you look into the albumin gene, that is precisely expressed in your liver cell, but crystalline is expressed in eye cells. You will not find crystalline is expressing in liver or lung cells. And there are various others. If you go to the lung, take a deep breath, your lung is not brushed. Because why? There are surfactant genes. Surface active properties are there. They are taking care of it when volume is expanding. So those are known surfactant A, B, and C genes. So precisely coming to the molecular level and quickly rounding it up. But this is the, what is this? Transcription bubble. Before a transcription start, it is the different kinds of enhancer is there, activator protein is there, RNA polymerase, and various other accessory proteins are there. So what is cancer? Undefined growth or unregulated growth. Various characteristics are there. Precisely when you look into the cell cycle, there is a G0, that is resting phase, then G1, S, G2, M phase, mitotic phase. G1 phase is largely 11 hours. S phase, that is synthetic DNA replication, occurs 7 hours. 5 hours is G2 phase. It's checking whether all the organelles are equally composed or not. And then mitotic phase, when it starts, it will finish by 1 hour. So, but in many ways, the cancer cells don't follow the universal rule. As a result, what happens is that cell is growing, you are initially getting tumor. When tumor is growing, equally, they are not getting food because of the scarcity of the blood flow. That's why new blood vessels needs to be generated, that is vasculogenesis. And when the nutrients are less there is a gene known as hypoxia inducible factor. And when oxygen supply is not there, blood supply is not there, cells are not getting oxygen. So hypoxic condition, that gene is activated. It has a tremendous curiosity to find out why the channels are stopped. It calls first VEGF, vasculoendothelial growth factors. Altogether with certain other molecules, it starts. At that time, you just remember the story of Mahabharata. Shakuni was only given all the foods from their fathers. Here the cells are getting all the resources, autophagy, etc. by managing their alive. Integrity lost, cell-to-cell integrity, that we call cell adhesion molecules are not expressed. And when blood flow comes, they are not ready. They don't know to swim. They just come. And this way, all over the body, it spreads. And that is known as cancer metastasis, the molecular basis of cancer metastasis. So, it is an endless process. I will stop here to give the space to other speakers. I will come to the end. I request the who is managing to come to the last phase of not only research I do, I have to teach a lot of things. In one photograph it shows the students. The 11th April, my birthday, the students were, I was taking class. At that time I was HOD also. A lot of works. So consecutive 2 classes. I was surprised by them. Sir, give us one just 10 minutes. They celebrated my birthday. So that is so the batch on that day. Laboratory alumni, PhD degree awarded so far 11 and another one has done, so 12. A lot of fellows trained, but unfortunately due to different types of constraints, they were not able to finish their PhD. Postdoctoral fellows are also there. Now one is working also. Guidance of research project, it is not 25 or 28, it is so far 64. Where there is no money, you don't can procure any honey. Research depends on money. So by God's grace, I am funded from the institute as well as DBT, ANRF and ICMR. Thanks to ICMR. So ICMR recognized my lab as Center of Excellence in the last year, 2024. And when you, without getting any money from your own work, own capability, people tell you you are an excellent boy, excellent girl, and excellent lab. It gives immense pleasure. So feel proud. So these are the current strength of the lab. 8-9 students are always there in the lab. Publications, you can visit the PubMed. Little bit are there just in last 4-5 years, whatever is done. Thank you. All depends on your neuronal network. As strong it is, so much you are, presentable, competitive, whatever you want to say. So think about it, and you don't try to overact him. It will give you a drum beating like that one. And our Mr. Madam will tell you guys about that one, that you don't try to excite more your ganglions and others. So thank you all. Nice in young days while working in UCSF. My mentor, he was Indian origin, and some of the lab members. I took only the UCSF part, and elsewhere and everywhere in the world when I visited, there will be history. No time to share. Thank you all. I know when I speak, it's a torture for you, those who don't like, and it's a pleasure for them who likes it. Thank you. Thank you again, all the stakeholders of this nice university. I'm really very much happy, and special thanks to my beloved friend. He's young, but he's a very good friend, your current Vice Chancellor, Professor Dandopat. Thank you all. And Professor Dinda, nicely arranging all these things, reducing the burden of his beloved Vice Chancellor. So, when you want to do something, nice faculties, nice students, all these makes a sense. Whatever I showed here, I cannot show you my pictures and other things. So I will come again to this university to talk about the subject matter of our research. So that will give you. But thanks to the PhD students, MSc students who developed the data. What I'm here standing for them as a good scientist. As a student, I did a lot, but that is not sufficient enough to get the excellence of ICMR counting. So that is credit to the students. Thank you all again.
Thank you very very much, sir, for enriching our understanding on the cosmos and the life from both chemical, physical, as well as biological perspective. Now it is my honor to invite our next speaker. A very dynamic speaker is present with us, Professor Bidang Das Mohanty, sir. Please welcome them with a big round of applause. Sir, we sincerely look forward to your insight. Kindly take the podium, sir. Thank you.
Hello, hello, hello. Thank you. Pointer. More slide. So, very good afternoon to all of you. I'm standing between two speakers and lunch. I'll try to finish. Let me start by thanking Utkal University for giving this opportunity. It is always great to come back to your own university. Thank you. So today is National Science Day. Okay. The theme is, as you have heard, I'm not going to speak on that theme, but I'll try to tell you about some interesting science which happened in last 10 years. I want it to be very interactive. So how many of you are students, masters and PhD? Can you raise hand? Oh, very good. So you will interact with me, huh? Then that I want to ask, where were you on 14 September 2015 at 5:51 a.m.? Huh? Kothilo? School re, ghare, of course. But satobale school re na, college re na, kouthi? Okay. Huh? So I'll tell you about an event which happened in 2015, 14 September at 5:51 a.m. Before that, let me tell you, this is National Science Day, and this is a portrait which is made by Homi Bhabha of C.V. Raman. I come from a DAE institute, which is, DAE is all started by Professor Homi Bhabha. So the title of my talk is When Space-Time Trembled. Before I go there, you have already heard about this. So this is Professor C.V. Raman. He got Nobel Prize in 1930 for his discovery of Raman effect. The government declared in 1986 as a National Science Day, and since 1987 we have been celebrating this as the National Science Day. Raman effect, the paper appeared in Nature, see that, and it's a very simple phenomenon I have shown here. You know, there was radiation scattering. So a light comes and the scattered light had the same wavelength. But in case of Raman, the scattered light and the incident light have different wavelength. And that wavelength depends on what molecule scatters it. And see how profound this effect was. Now you have direct applications. Applications across all fields of science. In fact, the machine named this effect, Raman spectrometer, one of which is going to come in Utkal University. Then you can really see this machine. What a marvelous journey since 1930. Okay, so let me come back to my talk. How many of you know Newton? All of you know. What is Newton known for? Kichi subuni jore koibo us class re koucho jore? Newton? Okay, gravitation is what I will talk about. So Newton is known for his laws of motion. This is widely used across mechanics, engineering, space science. Newton is also known about gravitation. I'll come to that. He is also known about calculus. You know the calculus which you read, he is a co-inventor of calculus. He is also known for optics. He is one of the biggest scientists of any generation. You agree? Newton. Laws of motion, gravity, calculus, you name it, he has it. Okay, good. So, And Newton matters today because every rocket you launch solves some Newtonian equation. Every satellite that you put in the orbit follows some Newton's equation. Every engineering structure that you build, you need calculus, differentiation, so it is there. Every physics equation you solve is mostly solving the force equations. So Newton is everywhere there, right? And somebody asked Newton that what is his contribution, and this is what is at. I do not know what I may appear to the world, but myself I seem to have been only like a boy playing on a seashore, and then finding a smoother pebble or a prettier shell than ordinary, while the great ocean of truth lied undiscovered before me. That is what he felt. So he has done so many remarkable discoveries, but he feels that like a boy, he picked up a nice good, he came up with laws of motion. Then further, and he picked up another cell, laws of gravitation. He picked one more thing, he found optics. But he still feels this ocean of truth like him. Okay, so you are convinced, huh? Newton is the scientist that we know. Not convinced? Convinced. Everybody saying yes. Okay, very good. Now I want to tell you how he got to this. It was 1665, and great plague occurring, and University of Cambridge was shut down. He was 23 years old. Then he went to his home. There was no laboratory. There was no professors. Of course, that time there was no internet. CERN came later. There was just solitude and curiosity. While everybody has seen many fruits falling, it was only he who asked why. Then he said, is the force that is also acting on the moon? Can you imagine? Why should I connect something falling on earth to something to the moon? But that's what he asked, and that is what he took. During 1665, people felt that whatever is happening on earth is very different physics from whatever happening in heaven. But it was Newton who told is the same gravity which happens on earth is also responsible for making moon move around earth, earth move around sun, and so on. Newton unified them. Now just think, few years back, just imagine the situation and just think, few years back there was COVID. Universities were closed. How many of us thought and have given such a profound idea? कौन ହେଲା? सेठी प्लेग थिला, एठी कोविड ହେଲା. You see, it's a remarkable, and he ruled the world for almost 200 years. Okay, and give a equation ta dekhicho na nahi? F is equal to G m1 m2 by r square. Masses separated by a distance r. The force of gravitation is given by this expression. This expression rules what happens on Earth, gravity, how planets move, satellites orbit, comets move, tides, all are governed by this Newton's laws of gravitation. Everybody agreed. 200 years, everybody agreed. And then Einstein came. Oh my God. He gave his theory of relativity. Hey, Banaputland. Hello. How? Einstein came. So someone Einstein gave theory of special relativity. बहुत जोरे फुटला. बाबू रे. So Einstein came And then he said, gave his special relativity and general relativity. According to special theory of relativity, nothing can move more than speed of light in vacuum. ये जो न्यूटन जो फोर्स इक्वेशन देले F is equal to G m1 m2 by r square, it is instantaneous action or force. That means as soon as two masses come, there has to be a force. That means signal must have traveled with infinite speed. But Einstein said, well, no, signal cannot travel more than speed of light. So there was a problem. Action at distance, infinite speed. So then he started thinking, maybe gravity is not this force that we know. Gravity, he told, is related to geometry. Gravity could not be a traditional force. So what is gravity? Dekho, this came in 1915, around 1915. Se porjanto jake, whatever Newton said was all accurate. Now also we are using Newton's equation. But what is really gravity? So Einstein said, you know, the space and time is like a rubber sheet. And when you put a mass on this rubber sheet, it will cause a deformation. It upar dekho deformation. When another mass is dropped, then it will roll towards that heavier mass. Okay? So Einstein theorized smaller masses travel towards larger masses because it is not attracted by some mysterious force, but because the space gets curved. Space and time gets deformed. Gote rubber sheet re gote heavy mass roila, gote choto mass ta pakeile, automatically because of the curvature, it will get attracted. Au jodi se choto mass ta ku tike speed re dabo, it will go on moving. So see how different it is. Nothing to do with the way Newton thought. It's actually space-time which is deformed, right? Now question came, so how to prove that what Einstein said is right? Okay? So, but just remember, gravity, according to Einstein, is not a traditional force. Mass and energy curve the space-time that we are in. Objects simply follow the curvature. Amara aguru idea thila, mass, force, motion. Einstein koile, na, mass, curvature of space-time and motion. He also gave his equation. Okay, setting jibani, complicated, it's a tensor form. So he says, Earth is not pulling the moon. Earth is actually curving the space-time and moon is simply following this curve. That's the idea. Pura silence Raji no believe, you don't want to believe it. Huh? Pura total silence. Okay, so 1915 they chandi silence okay, chalibo. So then, if that is the case, the key point kon thila, je mass thile, force habo. Photon is a particle which doesn't have mass. So if Einstein was right, then light should also bend because light the space-time re travel kurchi na. Jadi space-time bend he jibo, light bhi bend he jibo. So light bend, light ro mass nahi. Tal pura Newton jaha koithile se thik habo. Light jadi bend he gala, that means you know that what Einstein said is right. So kemiti janiba light bend hochi ki nahi. So there was a scientist Arthur Eddington. Se kon kale, he went to Ghana, Africa, that because there was a solar eclipse. So there was a star, okay, and he observed it. So he knows where the star is. Then he waited. Then there is solar eclipse. So the sun came in between. Star and he found star is there. Then sun came, moon provided eclipse, so pura dark. But because sun is a heavy object, the space-time got deformed. So now he cannot locate the position of star is there. Where? When the sun was not there. He found somewhere else. It is like a lens. Apparent position changed because light bend. Straight, dekhucha, kichi object majire nahi, space-time is not bent, you know the position. Now you bring a heavy object, which is
sun. Solar eclipse, baki light, so black hegala. But when he tried to observe the position of star, it was shifted because light had bent and light was showing a different direction. That's just simple lens. Lens केमती काम करे. He was so excited, she गोटे कविता लिखिदेले. जो conference को गले, she started singing this. What did he sing? Oh, leave the wise our measures to collate. One thing at least is certain, light has weight. One thing is certain and rest debate. Light rays when near the sun do not go straight. Light has weight. Light is supposed, photon is supposed to have no mass. Okay? So this was first proof of Einstein's theory is correct. You know, we are using GPS. If Einstein's, according to Einstein's theory, satellite re jo clock achi, our earth re jo clock achi, ta bhitere time difference is 45 microsecond per day. Jadi se factor taku correct न करा हबो ताले तमे भाबुछ उत्कल यूनिवर्सिटी रे अछ किंतु एक्चुअली तमे बरमुंडा पाखे. There will be 10 kilometer at least off by per day. Okay, so Einstein's theory is used so that the GPS is really accurate. Okay, today I'll tell you one more proof which took 100 years to discover, and that's why Einstein's theory of gravity is correct. He said, Einstein said, if it is true, space-time is a rubber sheet, tabla, okay? When you put an object, it vibrates. The tabla vibrate. Or you take water and you put a stone, it will also vibrate. Similarly, space-time must vibrate. And that wave is called gravitational wave. Jemti water re padla water wave, tabla re padla sound wave. Semti, this vibration, the wave it creates is called gravitational wave. So she koile maybe a wave ta jadi detect hei paribo. He was not convinced. Einstein 1915, he was not convinced that one can detect, but he said there should be a wave. Okay. Now, on a gravitational wave. Now, gravitational wave is something which shrinks space along one perpendicular direction as it stretches the space along other direction. It is just vibration, as you see. I'll show you it is like this. Space vibrates and generates wave. Okay. Now, how can it generate wave? Now, it can generate wave like this. Suppose two massive objects like black holes, they come and collide. Then it will generate ripples in the space-time. And those ripples, if we can detect, then we have detected the gravitational wave associated with vibration of space-time. Okay? And Einstein himself felt it will be very difficult to detect it. Okay? It took 100 years. In 2015, 14 September, it was detected. Okay? And that is what I want to tell you. And how the scientists detected it. Now, physics re jadi amar ete nicer UG lab, undergraduate lab re hochi, gote jinso achi, seta konde Michelson interferometer. Eta bodhe plus 2 re jadi science nei thibo relativity re bi padha hui. Plus 2 re padha hochi? Padha huni? Se tama padhautuva to, tame ene ko college re padhaucha? BJP college? Au? Acha. Kendo padhauchi nai ମୁଁ ପଢିଛି ମତେ ଲାଗୁଛି ଆଚ୍ଛା ହଉ ଠିକ ଅଛି. ଏଟା ଭାରି ସିମ୍ପଲ ଏକ୍ସପେରିମେଣ୍ଟ. ଏଠି ଗୋଟେ ଲେଜର ଅଛି, ସୋର୍ସ ଅଫ ଲାଇଟ, ଏ ଦିଟା ମିରର ଅଛି, ଆଉ ଗୋଟେ ସ୍କ୍ରିନ୍ ଅଛି. ଓକେ, ଏଇ ଲାଇଟରେ ଆମେ ଫାଷ୍ଟ କ୍ରିଏଟ୍ କରିବା ପାଥ ଡିଫରେନ୍ସ. ପାଥ ଡିଫରେନ୍ସ will create phase difference. Phase difference will create interference. If the two waves are in phase, it will be a larger wave. If they are out of phase, it will cancel. Okay, ये जो interference जो गोल गोल देखुछ, those are interference patterns. Okay, इटा UG student करूचंदी, एठी भी निश्चय BSc रे करी थिबो म. करनी ना फिजिक्स पिला नांती तुम जोर कहून डोमिनेट कर जाउछन कहउछन नहीं ओके सो एटा ना सब कॉलेज रे यूनिवर्सिटी रे होउची ए एक्सपेरिमेंट ये एक्सपेरिमेंट करिकिरी ग्रेविटेशनल वेव डिटेक्ट हिची ओनली थिंग हला ए जो मु कहूची से कौन कले से माने ए एक्सपेरिमेंट ता सेटअप कले जोठी की एमटी इंटरफेरेंस पैटर्न अछि ओके मने रख ए डिस्टेंस गुडा को इंपोर्टेंट डिस्टेंस से स्प्लिटर रु मिरर ओके ए डिस्टेंस ऊपर डिपेंड करिवो केमिति पैटर्न पाइबो से experiment the set so that you have a pattern. Now imagine a huge gravitational wave pass through Earth. What will happen? Now space time itself changed, so a distance change hijibo. Change hegale a pattern vanish korijibo. That is the signature. You set a experiment, pura stable experiment, space and time re, where you have a interference pattern. Now suppose some phenomena like gravitational wave will itself change space and time, then that pattern will disappear. Okay, that's the experiment. So you have gravitational wave experiment, you have the same Michelson interferometer, and then you wait and you so this was your, okay, let me put it there. Suppose this is the initial length. Gravitational wave passed, this length changed. It changed, but calculation कहला से gravitational wave जो pass करिवो, it will change the length by 10 to the power minus 18 meter. 10 to the power minus 18 meter. Realize करते सेटा कितने बड़ा, बड़ा ना छोटा? छोटो अच्छा हो रियलाइजेशन पाई गोटे कहूची जस्ट टू टेल यू ये हला 1 मीटर इज 40 इंच ओके ह्यूमन हेयर 10,000 गोटे मीटर रो 10,000 टाइम्स कम छोटो 100 माइक्रोन ओके आउ ताको डिवाइड करबा 100 थरो हला वेवलेंथ ऑफ लाइट आउ ताको डिवाइड करबा आउ 10,000 थरो एटम रो साइज पुणे आउ डिवाइड करबा आउरी 100,000 times nuclear size. तो हमको आउरी 1000 गुना sensitivity होगा दरकार. You should be able to detect changes 10 to the power minus 18 meter. These are some of the smallest measurements human has made. So they waited. से experiment में किच्छी मिलनो थला. They upgraded the detector which can have sensitivity of this order. And then suddenly they found gravitational wave. केमती आसिला से इंटरफेरेंस पैटर्न अछि लेंथ चेंज ही गला ग्रेविटेशनल वेव जेतले पास हला पैटर्न वैनिश ही गला ओके ओके लेट मी सो एक्सपेरिमेंट कोठी होतला इन यूएस देयर आर टू फैसिलिटीज जो इंटरफेरोमीटर जो लेंथ कहली से लेंथ गुडा का 4 किलोमीटर वन इज एट हैंडफोर्ड another is at Livingston. Dita independent जगह होतला. This is how. ए इंटरफेरोमीटर से जो लेंथ ता देखी थी स्लाइड रे ये हला ए 4 किलोमीटर सेपट आउट 4 किलोमीटर ओके that huge facility this is at Hanford and this is at Livingston देखो तो dita line okay now ए जिंसटा को करिया पाई you need lot of technology, engineering support. Without this, it is not possible. Simple, I have listed what are the requirements. For example, there are mirrors, na? Mirror to light reflect huji, Michelson interferometer. The mirror has to be very smooth, smooth at atomic scale. There cannot be roughness of the level of atomic scale. So imagine the technology which has to be developed to make those mirrors absolutely smooth to detect 10 to the power minus 18 meter changes. Then there are other things which can cause the vibration like earth is constantly vibrating, noise from nearby truck, ocean, earthquakes, all these has to be suppressed. they really have to make it vibration free, the setup. And only when space and time shrink and expand, they should be sensitive. That's the challenge. I've listed them here. It really required mastering of lasers, material science, vibration control, quantum optics, everything at once. And then what they saw. ମୁଁ ତାପରେ ଗୋଟେ video ଦେଖେଇବି, then I think I'll be almost done. On 14 September 2015, they saw two black holes. One black hole mass is 36 times mass of sun. The other was 29 times mass of sun. They came and merged. They merged to form 62 solar mass object and released, se jo merge ra impact hela, they released energy 3 solar mass. This energy in form of wave traveled. It traveled 1.3 billion years to reach this experimental facility. And the signal stayed only for a fraction of a second, 0.2 seconds. Okay? And it caused length changes of the order of 10 to the power minus 18 meter, or strain changes of the order of 10 to the power minus 21 meter. And that's when we as humanity heard universe, huge objects in universe collide. And that was the detection. Okay? They publicly announced it on February 11, 2016. I will now show you a video. And if you really make pin drop silence, that energy has been converted to sound. And the sound that you will hear now almost corresponds to 1.3 billion year old universe sound when two black holes collide. Just think for a moment how profound is this, that we were able to detect something colliding in this universe 1.3 billion years. The sound will last for 0.2 seconds. So you have to be really silent. Huh? Okay. Sir, you play karibe. Thank you. 1.3. These are the two black holes. They're coming, slowly coming closer and closer, and they will merge. It will lead to gravitational wave. This is a simulation of how it happened. The waves passed. It reached the experiment, LIGO. So what did you hear now? Isn't it amazing that we are able to hear the sound, energy converted to sound of two black holes which collided 1.3 billion years ago, validating Einstein's theory of gravity, telling that Newton's laws of gravity, action at a distance with infinite speed, is not right. It's the space and time which is curved, and that leads to gravitation. Eddington has shown it for light, and 100 years, because we had to reach that technology, after 100 years, we showed the discovery of gravitational wave. Of course, the whole world noticed it. And of course, the persons got Nobel Prize. 2017, the three heads of those experiments, it's an experiment of many people, were awarded the Nobel Prize for their decisive contribution to LIGO detector and observation of gravitational waves. And you as young scientists have great opportunity to participate in LIGO India. India has now an approved project to set up the same experiment in Maharashtra, in Hingoli. Okay? We got approval in 2016, funding confirmed in 2023, and science should start in 2030. Okay? That is where the setup is. How this is going to aid? Now, if you have three different, there are two in US, one in India, of course, there is also one in Europe. This will precisely tell you where that collisions happen, a black hole or neutron star. Okay, this is my last slide. Okay, two slides more, but this I want to tell you again for the younger people. This is Big Bang model of how universe evolved, okay? If you are, it is up to you which part of the evolution of universe you are interested in. The government of India, Department of Atomic Energy, Department of Science and Technology is going to fund you to pursue, to know how the universe evolved over 14 billion years. If you want to know about microsecond old universe, you can join CERN. If you want to know how fast nuclei were formed, Facility for Antiproton Ion Research in Germany. If you want to know how the first stars formed, 100 million year old universe, square kilometer array experiment. If you want to know how the structure of this universe happened, 500 million year old universe, 30-meter telescope. In between, we have our own GMRT, which is located in Pune. If you want to know how fusion happens, how energy is produced in stars, ITER. And if, of course, if you want to know about black holes and gravitational wave, LIGO. There is a link there. It gives you vision document of Indian science in nuclear physics, particle physics, astrophysics, accelerator. 4 documents are available till 2035. This is from the Principal Scientific Advisor to Government of India. And I was associated in writing that orange one. And then there are other 3 documents which are available. Please look at it, what science we will do in 2035, up to 2035. I will end by giving a quote which Sir C.V. Raman told after his Nobel Prize in a college in Madras. And this is very important for you all. I am quoting him. Youth is the most glorious time of all. I have said elsewhere that most of the great discoveries in science have been made by young people. It is not the experience or wisdom that the old age bring, but the freshness of outlook, the indomitable desire to achieve, which is the characteristics of youth that makes discovery possible. It is this that makes life worthwhile. If only you realize this, and realize that here I am still young, let me see what I can do, that all discoveries become possible. Don't forget this. Very profound, and the future of Indian science lies in your hand. And that is because Newton did something when he was 23 years, Einstein did something when he was 26 years, and Raman did something when he was 39 years. So you are not too early, you are not too late. Perhaps the next Raman, next Einstein, next discovery will begin here. Who knows? It is your responsibility to do that. But in doing that, do not forget that the ocean of truth is still undiscovered. It is waiting for someone in this hall. Happy National Science Day, and thank you all.
Thank you very much, sir, for igniting the scientific curiosity among the students which are present here with us audience. I think that it was with your lucid and clear presentation, you have reflected the true spirit of National Science Day. Thank you very much once again, sir, on behalf of Utkal University. So moving on, we are now honored to invite our next speaker for today, Dr. Sushil Kumar Singh, sir, scientist ICMR RMRC, who will be sharing his expertise with us on the topic, the long fight against malaria, patents, innovation, and global hunt for the vaccine. Sir, I request you to kindly take the podium.
Thank you so much, Honorable Vice Chancellor, Utkal University. Sir, thank you so much for inviting me. I am really feeling great that in front of you and other two speakers who spoke a very nice, science. So I'm feeling like I'm a child here in front of them. But I will try my best to put our science what we generally used to do at RMRC Bhubaneswar, as well as what I did previously in Copenhagen University. But mostly because I'm coming from RMRC, so it is my responsibility also to tell about ICMR and RMRC is doing, so then we can collaborate each other. So uh, in a National Science Day, first of all, I will say namaste to all women, and I will dedicate this Science Day, Women's Science Day, to three of the women. One is my mother. Because of her, I'm here. I become a scientist because of her only, and she is only class third pass. In her time, she was class third pass. And another woman, my wife, she is IITian, but for me, she is serving home. She did PhD from IIT BHU, but now because of me, she is just doing all the homework, also participating in science with me. And third woman, because of her, I am in a Bhubaneswar. It is Dr. Sangmitra Pati Madam. So I dedicate to her as well, and she is my mentor. And whatever work facility we develop here because of her only. So okay, now I will start with my presentation. So today's talk, what I would like to tell you about is a long fight against malaria, patents, innovation, and the global hunt for a vaccine, because I am used to make a vaccine, not only for malaria, but other disease as well. So, but before I start my main topic, I would like to tell you what kind of facility ICMR used to give for a scientist and for a collaboration. So here, if you can see that ICMR have a very big infrastructure, because ICMR is a institute of more than 100 year uh, old, and it have 28 institutes, including headquarters, so 26 functional institutes, and we have very huge infrastructure. You can maybe take a photo of this website, and if somebody need any experimental expertise things, you can contact any scientist anywhere in the all over the India. And I am also a nodal officer of medical innovation at RMRC and representing ICMR. So I also have responsibility that I should promote a patenting and technology transfer. In my research career, I did four technology transfer. Two of the malaria vaccine is already in clinical trial in Africa, and I will tell you what we did here at RMRC and which is going in a clinical trial in future in the later slide. So we have a patent mitra. Anyone can log in here in patent mitra, and if you want any support for doing any patenting things or technology transfer, patent mitra is there to support you. From RMRC Bhubaneswar, I will be there to help you guys, and it is almost free of cost. So like DBT have Bharat to help you, so similarly ICMR have patent mitra and medtech mitra to help you for doing all the technology transfer and things. And how patent mitra can support you and where you can support you. This is patent filing, like this patent filing, patentability assessment, patent prosecution support, technology transfer and capacity building, ICMR and medical college institutes, startups, and any institute led by biomedical innovators. Anybody can take help from patent mitra free of cost. So this is just a picture from internet that how India is evolving in the patent. So you can see that since previously we had very less patent filing cases, but now it is increasing, and you can see that in 2024 more than 1 lakh patent has been filed. So we are also started following others to make a patent. And I can tell you one thing that I met a patent officer at Delhi, and he said that we are very hesitant, always hesitant to submit the patent. But mindset should be whatever you do, first of all, just fill the patent and then publish. But what we do, and as academic persons, we used to publish a paper, not to patent, but change your little bit mind and then start patenting, like submit today, tomorrow you can publish, there is no problem. So that kind of attitude we need to develop in a science, then we will, we can complete US or Europe or China. We are well behind from them. So, so now I will tell you that what RMRC is particularly doing and how we can help each other. I mean Utkal University and other who are listening here, that how we can collaborate and how we can help each other. So not moving. What happened? It's not moving. It's, yeah. Okay. So ICMR is doing, RMRC is doing malaria and phalanxis research since long. So here recently we developed a multi-stage malaria vaccine for falciparum, and also working on buyback, that I will tell you a couple of slides later. And molecular detection tool is also developed at RMRC, and improved MDA program is ongoing. RMRC is also participated in a Daman program for elimination of malaria, and it was a big success. More than 82% malaria was reduced during that time, but unfortunately COVID came and then detection stopped. So again they are starting Daman program. And also we are doing a lot of surveillance, like we have a BRDL for disease, all the viral surveillance, and we also have a national reference laboratory for tuberculosis. We are heading for 10 states. AI-based TB screening program also we are doing. We developed AI method for detection of TB. Cholera monitoring is ongoing. Tribal health, we are doing a lot of work in the tribal health, like sickle cell screening. A new G6PD variant discovered. Nutritional program is ongoing. Maternal, child and health is also a focus of RMRC Bhubaneswar. And also we did a great work in the COVID-19. I don't need to tell, but everybody know RMRC after COVID only. But I have a story. I came before COVID, and I was in the airport. I asked, where is RMRC? Nobody could tell me where is RMRC. I was in a Nalco Square. Then I realized that it is RMRC. Otherwise, from airport to Nalco Square, nobody knew. And now everybody know RMRC. As well as Dr. Sanghmitra Pati. RMRC, Sanghmitra Pati, both are now synonymous. Everybody know them. So, and we also have, MPH program and PhD course through ACSIR. So now ACSIR and ICMR came together and make a university. It is called ACSIR. So this is the infrastructure we developed for vaccine development. So this is my lab in general. We developed in 1.5 years. So all these molecular biology, protein chemistry, and immunology, all the work one can do. We can collaborate anybody. And also we develop recombinant protein production platforms. So we have 4 platforms: E. coli, Pichia pastoris, and most importantly Lactococcus lactis. That is a grass, and it don't secrete any endotoxins. So this is a very, very nice platform for producing any kind of protein production as a soluble protein. And we are also developing this Biomax Mori-based eukaryotic system. So now I come to my original topic, malaria. So everybody, most of you have already know that what malaria is, so I don't need to go to the basic about malaria. But for setting the presentation, I just wanted to tell you that the malaria is a very, very complex parasite. And you can see that you have 2 main hosts, mosquito. This side is a mosquito, and this side is the human. So making a vaccine against malaria is most difficult task. I will tell you the story how people started working on malaria vaccine development, how long it took. it took decades, more than 50, 60 years to make a vaccine. So I will tell you the story after that. So here, if you see, even in our Ayurveda system, in a old ancient Atharveda and Charak Sahita also mentioned a fever like a malaria. So since then, our ancestors know that there is something malaria like fever is there, and it is mentioned in Atharveda and Charak Sahita. But in the laboratory, scientific discoveries started in 1980. So first of all, in 1980, parasite is discovered, and then in 1994, vector discovery happens, and then transmission blocking discovery happens, and then first medicine came as a chloroquine, and then chloroquine resistant also happens, and then whole sporozoite immunization happened, then another, because of artemisinin is resistant to, then artemisinin discovery happened in 1972. After that, people started culturing parasite in 77, and then full-length CSP is sequenced. So this is the 1984. From here, my journey started. Although I born in 1980, but my journey started from 1984. So when first time the CSP protein was detected for a vaccine candidate, then RTSS came into the picture. You most of them know, if you know the malaria, that there is a two vaccine. It is one is RTSS from USA. It is GSK company who made this, and then other is R21 made by Oxford University. Fortunately, both of the vaccine is being produced in India only. They discovered in the US and Europe. A production is happening in India. So why I am pushing a little bit about this full-length CSP? Because both the vaccine is based on only full-length CSP. It is based on the CSP. So in 1987, the trials, RTSS trial started, and then vaccine approval happened in 2021. So you can see that how long it took. First of all, when trial started in 1987, and then it took around how many years? 30 years to approve the vaccine. But the discovery happened in 1984. So now it is around 40 years. And then in 2023, our vaccine came into the picture. And I will tell you what is the difference between the vaccine which took 40 years and how our vaccine is better. So why I was telling that this 1984 my discovery started, because at that time I was 4 years, I don't know anything, science, anything. But based on that CSP, we got patent in 2023. So around 40 years, there was a number of patents in this protein, but it was difficult to produce full length and properly folded protein. So that patent I got for producing. And I was feeling privileged that I was the same scientist who discovered CSP in 1984. So before I'm going to tell our story, you can see the current status. What is the current status of the malaria globally? So you can see here, this is Africa. It is totally red. And whole Africa is almost endemic. People are dying, mostly 0 to 5 year old. But what is the scenario in India? India is now becoming, low risk populations. So India had very good program for controlling malaria. So now, slowly since last 10 to 15 years, we are reducing the malaria burden and becoming almost very low risk. And maybe it might take 10 to 20 years to become malaria free if vaccine and other supporting materials start working. So what is the challenge in the malaria case? So challenge is nothing but, you can see that Plasmodium have very complex life cycle because you have two hosts, human and mosquito. And if I tell you in Hindi, there is one word, I don't know what I should tell in English, but it is called Bahurupia. I don't know how to explain this. Bahurupia means in one stage it is a different size and its structure is different, protein is different. When it go to another stage, protein is different, its structure is different. When it's in transmission blocking, in a sexual stage, parasite is different. So in all the states it is totally different. And so and protection is also very low. Although in India, number of people are exposed to malaria, still they get malaria. So protection is not there. Drug resistance is also increasing day by day. Climate change and vector expansion is there. Vaccine performance in different uh, uh, transmission setting is also uh, difficulties. And the most important thing is country like India or Africa, manufacturing cost we cannot afford. You should have a very, very low cost vaccine, because the two vaccine, malaria vaccine, which is already WHO approved for uh, selling, are costing, you know, how much? Minimum $8 per dose. So meaning 800 rupees per dose, and they used to give 4 doses. So our trick, or to 3,200. I don't think anybody will take it in a remote area, and government is not going to drive this 3,200 cost vaccine. So that's why a cost-effective and the cold chain logistics should also be very important here, because in India, in North India, you have sometime 5-degree temperature, and South India 30-degree temperature. Temperature is very variable, so we need to have something which can be stable in room temperature. So that. Sorry. So thinking all about this, we started working on making a vaccine which could be useful for Indian citizens. So here, this is our vaccine model, what we do. So if you understand that when mosquito bite, so here you can see this number 20 and number 10. So focus on this only. So when mosquito bite, sporozoid goes into the liver, and only 20 sporozoids survive. So there is one bottleneck where you can catch the parasite. And after this, when it go to the blood stream, there is a number of parasites, 10 to power 11 parasites. So that time it is very difficult to kill them. And also because it is inside of the RBC. So RBC cannot be killed by your host immune response. And then during this asexual cycle, around 10, again when mosquito bite, 10 parasite male and female gametes used to go to the mosquito midgut. That is also one bottleneck where you can catch. So what we did is, we just catch both the bottlenecks. So if you compare with the two best vaccine, that is RTSS 21, so they are just this much, the very small part. They use this very small part, you can see blue line. And what we did is, we took full length CSP and then a transmission blocking vaccine candidate. So this transmission blocking vaccine candidate is already in a clinical trial in the second phase in Europe, made by myself and my group in Copenhagen. And this CSP is also, we made a full length, and now we couple both of them and made a multi-stage malaria vaccine that is helpful for blocking infection in a liver, and also transmission blocking. So see here the difference between our vaccine candidate and RTSS and R21. So when a person is immunized with the R21 or RTSS, only individual can be protected, not the community. But when you immunize our 8 falsivax vaccine, then individual will be protected as well as community will be protected. So that is the need for India, because if you are immunizing one person and only one person is protected, but other community is not protected, then you cannot eliminate malaria. So that's what we did is, we made a multi-stage malaria vaccine, and that is protecting individual as well as community. So these are some novelty what I did. I don't need to read this. And then advantage over other. So most important thing is, for natural normal vaccine, you need to have a cold chain. What we did a formulation with the LM microparticle. With the help of that LM microparticle, our vaccine is active at room temperature for 12 months. So even you just make a vaccine, put room temperature for 12 months, it will be totally active. So that is a great achievement compared to other vaccine, because you need to have a low cold chain for other vaccine. That's why our vaccine become cheaper. And this is just a result for vaccine, how it looks. So after producing, we have a very nice production protocol, and it looks like this. And we can produce 20-25 milligram per liter, and all is already set. And we already did technology transfer to the 5 companies, 5 big companies. And I think it is first time from RMRC that for one technology, 5 big companies took license. And they are very big company like Panacea Biotech, in uh, Indian Immunological, and Idus Life Science, and Tech Invention. The big companies took the license, and they are going to invest around 1,000 crore for developing this vaccine. So these are some results that we are getting 90% protection and 100% transmission blocking. And similarly, because I believe that if you want to eliminate malaria, you need to make a vaccine against not only falciparum but Vivax as well. Without Vivax, it is not possible, because even in our Odisha, we have Vivax and falciparum neck to neck. So we need to make vaccine for falciparum, that, but Vivax is equally important. So that's why we have started, 2 years ago, we started a vaccine development for Vivax as well. A similar protocol, each and everything is similar, and it is also going to be clinical, in a clinical trial very soon. So apart from that, what we do is, because all these 2 vaccines which I talked about are subcutaneous needle-based injection. So now we are also started working as an oral vaccine, the same multi-stage malaria vaccine through oral route. So here we use 2 different platform. One is spore forming lactococcus, Bacillus coagulans, and non-spore forming lactococcus, because oral vaccine need to be protective from your pH. So that's why these 2 experiments are ongoing, and hopefully in 1 year this project will also give us some insight about oral immunization of malaria vaccine. And these are some advantages of oral vaccines. And apart from that, we are also working in a VLP technology. It is because you might remember during the COVID time, when you get vaccinated with the COVID vaccine, after 3 months you don't have any antibody. It is because the formulation was not very immunogenic. So that's why generally viruses are very immunogenic. So we are making a virus platform where you can couple your vaccine candidate and immunize. So directed immune response can develop. So memory cell and other things can be developed. So one of my PhD students is sitting here, Sunanda, who is doing this virus-like particle platform development, and we already proved that when you couple your, and this is also very exciting things. We are working with the Kari, our Ayurvedic department, where we selected some candidate on the basis of Vata Pitta Kapha, and we are doing mitochondrial genome analysis, because mitochondrial gene used to come from only mother. So after maybe sequencing with the genome, mitochondrial genome, and comparing with the Vata Pitta Kapha, we will find out the longevity of the person life. And I'm going to finish in 1 or 2 slides more I have. So for the researcher who want to collaborate with us at RMRC, who have the opportunity to use our platform state-of-art facility for producing any kind of proteins. And also we have around 50 antigen from Plasmodium falciparum covering all 3 stages. One can use that as well for any seroepidemiological and other studies. We also have around 15 antigen from Vivax, and we have very good experience with the technology transfer, and not only for India but international companies as well. And we are 2 vaccines already in clinical trial, and 3rd is GMP production. Vaccine development against Plasmodium Vivax is ongoing. We also have a lot of resources for sera and other things. One can use this sera for developing any assay if they want. Apart from that, if PhD and postdoc if want to interact with us, if they want to learn something or they want to participate, they can have opportunity, especially in malaria case. So immunology and antigen discovery they can have. We need a very good scientist who can help us in a cholera protection, antigen discovery, mechanism of waning, and also next generation adjuvant thermostable formulation and low cost manufacturing, these kind of things we can help each other. One of the programs I wanted to stress upon is where ICMR RMRC is having online certificate course on One Health. So one can participate in One Health program. Dr. Devdatta Bhattacharya is taking care of this program. So you can check the website, and if you want to register yourself, you can register. And most important slide, this is the most important slide for me, because I believe that if you are doing any science, the people who are participating indirectly have equal, they also participate equally, and we should give equal credit to them as well. So I believe that they are both scientists and other people who are supporting a non-scientific group should get recognition, similar recognition as a scientist. So you can see here the clinical, when you do clinical trial, volunteers are there. Without them, you cannot do any clinical trial. Advocacy group is also there. Regulatory and ethics specialists are there. Finance and administration. My regard to finance and administration as well, because without them, it is not possible. Scientists, administration, and financial department, all three when work together, then we can do any science, otherwise we cannot. Logistic and the manufacturing pupils, and also the communication expert, who spread the communication, who convince the pupils, they are also very, very important. So that's why this is the very important slide for me, that if whatever I made, because of them only. And then, now but last week, this is my team. It is a little old picture, but some pupils are also we added. And this is my motto in the life. When I do any research, I do like this. So thank you so much for listening, and thank you, sir, for inviting me. I'm thrilled. Thank you.
Thank you very much, sir, for enriching our understanding on the research work related with the malaria, which is being undertaken by ICMR RMRC. Thank you once again. And now, after listening to the three eminent scientists who have enriched us with their knowledge and experience, it is time to invite and welcome on stage a distinguished woman scientist. I request Dr. Priyadarshini Mishra, Madam, to kindly come forward and address the gathering. Madam, please.
Good afternoon, everyone. And I know, being a physiologist, right now what is happening in your body. I'll try to be fast, finish up. I would like to start by thanking Dandapat sir for inviting me here, but my profile sounded better when Chinara madam was speaking. I didn't really believe it. For me, when sir said you come and speak, I had no idea what to speak about, because I don't have this kind of achievements. So what I thought is, I would talk about from curiosity to connectivity and some of the life lessons which I got during this part of my journey. So from curiosity, small city, small town, big dreams. I was inspired by one literature book which was on Albert Schweitzer. Have you heard of Albert Schweitzer? Okay, he was the pioneer of humanitarian aid. And one word stuck with me, reverence for life. Reverence for life. You may do a lot of research. Fine. Remember your subjects. Reverence for life. And that stuck with me. And the early research part, if you won't believe it, my maternal grandmother, I saw her, I used to stay during holidays at my aunt, uncle, Nana's place. So what she used to do, she is not very educated, but when my uncle started learning dissection, she said, okay, I'll make you practice at home. So frog was anesthetized using gudaku. How many of you know gudaku? Okay. So the frog was anesthetized using gudaku. Then an old rubber slipper became the dissecting board, and pins were used, and I saw my first dissection there. So that hooked me onto science, and from that time till now, okay, I won't say I was determined. Only one thing was there in my mind, yes, I want to become a doctor. But science, never I thought that I'll enter into it as a researcher or something, right? So how many of you have those moments where you are uncertain that, am I doing it for just the heck of doing, or am I in the wrong field? Raise your hands. Yeah, that doubt should be there. Don't worry. Sometimes life will take you through these parts, and uncertainty is a part of life. So that is what I learned, is what shapes us are not milestones, but questions that we choose to pursue. It's not what we say, okay, I crossed this, now I have this in target. Sometimes it's okay to be uncertain, right? And the first uncertainty was the biggest question came to me, physiology. Okay? So I was fresh from MBBS, the best graduate of my institute with multiple gold medals. I thought I will kill the world. I'll be a big clinician. And then life happened. Okay? So I got pregnant with my first kid. I was set to join dermatology DNB in Mumbai. But I was forced to compromise and take up physiology. I'm sorry to say this. And this is a statement I'm not very proud of. Okay? So it felt like I was compromising. So initially when I joined physiology, it was with deep reluctance. Deep reluctance. But it felt almost like settling for something inferior. Okay? And when I joined there, I was all cocky with my attitude, you know. Okay, I know a lot. What is physiology then? I can do it without thinking. With one part of my brain, I can manage it. And then the reality stuck when I could not answer even a basic question asked by my teachers. That day, I got the thrashing of the century. And with deep humiliation, in tears, I started studying physiology. And the whole world opened up. The questions started arriving. Once I started reading, I said, okay, there are no perfect answers. It's all questions. If I go to one level, it's still becoming more murkier. The more depth I read, it became more unclear. And that led to a deep love and respect for my subject. Okay? So it's not like in life, the path you select is most perfectly suited for you. Sometimes the determined path may be the best one for you. How many of you have felt this at some point of your life? It's not a compromise. So I take back my words. It's not a compromise. It's the best thing which happened to me. Okay? And I will start off with the question or with a thing which all of you will do tonight. That is sleep. Right? So my journey started with asking questions about sleep. You know what? You feel hungry, you can know. Right? ज़्यादा से ज़्यादा कितना देर रोक लोगे भूख? 1 घंटा, 2 घंटा. Then you'll rush to the canteen, right? But sleep, you guys, I know you guys. How many of you sleep on time? 2, 3, 4, 5. Okay? Out of a crowd of almost 70, 80, 5 people are sleeping on time. Now let me tell you, this Sleep deprivation is one of the most disastrous thing you can do. The amount or the way you sleep in your 20s and 30s is going to influence your health in 60s and 70s. Sleep is a cushion which we all compromise because we can get away with it. Am I correct? I can see Netflix till 2, 3 a.m. and still get up at 8 o'clock to attend the class. But in the process, I lost out few stages of sleep, which were important. Why important? Because your chronotype, your daylight influences, you are genetically clocking yourself. Your chronotype is genetically determined. If you go against it, there is a chance that you will fall ill. Second, most of your memories, skills, learning, whatever you are doing in classroom is consolidated in sleep. So just a partial night sleep deprivation, a partial night sleep, one single night sleep deprivation, if you see here, P300 is an event-related potential which we check by putting electrodes over the head. Now, here it determines, it shows your executive function, that is your alertness and your level of reacting to an unknown stimuli. Just one night of partial sleep deprivation can reduce it. So imagine you are going to exam. By not sleeping, you have already affected your cognition. Okay? If you see this brain, the darker portions are the portions of the brain which are active during sleep. So sleep is not a passive process. So do not think that by compromising sleep, you can reach places. Okay? So with this message, rest is not passive. It is when you become what you are trying to be. So take time out to sleep. Now from here came the question, if sleep is disturbed, what happens? So we looked into the role of sleep in epilepsy. Epilepsy, you know that it is a seizure disorder where they have different types of seizures. Now what is a seizure? It is a synchronized electrical stimuli to the brain, sort of a shock to the brain. At one time, all the neurons are firing together. Okay? And many seizures happen during sleep. You know, we have three stages of NREM sleep, that is non-rapid eye movement sleep, and one stage of rapid eye movement sleep. Your body has the inherent capacity to prevent epilepsy. How? Because when you go into deep sleep, we get the delta waves which are synchronized. If you go deeper, you will land up with a seizure. So those who are prone to seizure, if too much of synchronization occurs, you may land up with a seizure. So what the brain does to protect itself, it has this REM sleep, rapid eye movement sleep. Almost similar to waking, but you don't wake up. But all these parts of your brain are active during REM sleep. If you miss out on REM sleep, there is a chance that a seizure will be predisposed. I'm not saying in everyone, in a person predisposed to epilepsy. Okay? So that relationship between sleep and epilepsy is very important. Okay? And what we found out is that loss of sleep, this is a condition, sleep apnea. How many of you have heard about OSA? Obstructive sleep apnea. Obstructive sleep apnea? Okay. OSA is a very common disorder which is there in many people, but we are not aware of it. It occurs in people who are snoring. Okay? Snoring and suddenly they get up. Have you seen such people? They'll be, and suddenly they get up. That means they have stopped breathing for some time and it is hypoxia. So your brain detects hypoxia and you wake up. Breathe again, go back to sleep. But it has disturbed your sleep for some time. So intermittent sleep deprivation, intermittent hypoxia. So this OSA can cause changes in proteins, certain proteins in your brain like amyloid beta and tau proteins, which should be removed, but they accumulate. So sort of a neurodegenerative change is occurring with OSA. And if you know someone who snores, please make them aware they should go for a sleep study. Okay? Now, if OSA is there, epilepsy can be drug resistant. Sometimes just treating the OSA can cure drug resistance in epilepsy, which is a very important link between OSA and sleep and epilepsy. What it means for you, poor sleep isn't just tiredness. It is a slow accumulation of neurological risk. So treating sleep disorders maybe one of the most powerful things we can do in long-term brain health. Keep it in mind, right? So this is what we saw. I'll just keep this. And from there, from sleep influencing epilepsy, we next went on to look at the gut-brain axis. All of you must have felt butterflies in your stomach. Have you got butterflies in your stomach? Or that sinking feeling, gadbad ho gaya, but you feel it in your abdomen. Yes, your gut is connected to your brain through multiple ways. Through the vagus nerve, through the immune pathway, through the microbiome in your GI tract. So multiple microbiome in your GI tract can produce a lot of substances which may be good if you have a good sleep, which may be bad if you have some kind of disease, addiction, something else. It will create lots of problems for you. So this group of organisms which is residing in your GI tract can influence brain. Okay? How it can influence? It can influence cognition. So what we looked at is, does the microbiome in OSA patients differ from that in healthy normal controls? And we could see that it is quite diverse. If you see here, the orange ones are the controls and the blue ones are the OSA patients. So their microbiome is completely different from the control. So something is happening because of the intermittent hypoxia and the sleep deprivation in OSA patients, which is changing the microbiome, and that is directly reflecting in their vigilance scores. So their cognition is also getting affected because of changes in the microbiome. So a dysbiosis may produce this. Similarly, I have also worked with ketogenic diet in epilepsy. So where drugs fail, a diet works. Can you imagine this? These children, I will say children, most drug-resistant epileptic are children. Very small kids having 20 seizures per day. Can you imagine having 20 shocks to your brain per day? More than 20. So much of issues. Their development is stopped. Their cognition is affected. Everything. And these children on 4 to 6 minimum anti-epileptic drugs, they respond to a diet. Though I knew about this, it still felt like a miracle when the seizures reduced with keto diet. It still felt like a miracle. And the happiest moment for me was when one of my children went for a trip to Bhutan without any issues, had a good trip and came back. That was my best moment, if you say, the mother saying happily that, okay, I took him to Bhutan and we came back without any seizure episodes. That was a beautiful moment for me. So, one mechanism by which ketogenic diet may work is through the microbiome. So we have explored at different time points of the diet whether the microbiome has changed or not, and there are some changes, but till date it is difficult to say how manipulating the microbiome will influence epilepsy. So this was the, if you see here, these are the patients, individual patients, and you can see the reduction in the seizure episodes. So you can see one child who had so much of seizures and it reduced to almost less than 1 to 2 per day, and also the severity also reduced. The severity of the seizures also reduced. Similarly, alcohol, another example where, okay, I will not say this, but please stay away from it. It can cause neuroinflammation, which will increase the permeability of your gut and lead to multiple changes in your brain. So an alcoholic is not alcoholic only when he is drinking. If you change the inflammatory status of your body, it will affect throughout the day. So I would advise brain ke saath khilwad nahi karo. You see, so many substances cannot pass through the blood-brain barrier. You know that, right? There is a blood-brain barrier. That's why when you eat a pizza, you do not fall sick. Lot of sodium cannot enter at one go to your brain. So there is a blood-brain barrier. But sadly, alcohol crosses. Alcohol crosses, so better not start. Okay, you don't know where it acts. So, if I see the gut and the brain, they work together. Your gut is healthy, your brain is healthy. That's why if you see the Ayurvedic texts, they advise something in, I saw in Charak Samhita, there is one line, Pathi. They advise the diet along with the treatment. So your gut can influence your brain. And the life lesson from here, the brain does not function in isolation, so why should we? As scientists, let's collaborate. I see this tendency of, ye mera hai, isme ghuso mat, this is my area. Have you faced this? Science does not progress like this. Idea le liya, le lo. This brain can generate a lot more ideas. दे दो, क्या फर्क पड़ेगा? है ना? छुपा के रखने से कोई फायदा नहीं होता है. The more you discuss, the more you talk. चाय पियो, अच्छे से discuss करो. अच्छे ideas तभी आएंगे. Okay? So brain networks from there to EEG. If the brain is influenced, the brain functions in not in areas. So here I will just add, the sir from Niger, he said that young brains generate ideas. Young brains are also before 25 not completely myelinated. You will take risks. So young brains, good ideas. Mature brains, perseverance. So do not give up. Just because, oh, I'm old now, I cannot do anything. You can do. Okay? So some research may start at 40, that's fine. That's fine. You have a more mature brain, a more perseverant brain. Okay? So we looked at EEG in depression, and we could successfully classify depressed brain through networks using machine learning. And that was a revelation for me also. I never knew machine learning was so difficult and so many layers to it. So if I say now random forest, I at least know something about the depth, the trees, the branches, and all those things. So the questions the reviewers asked were so tough that I had to literally go back and look into what I have done. Okay? So that was a revelation, and EEG signatures are there in every disease. So gradually we are looking into those things using EEG, because MRI, fMRI are little bit costlier. And then we also developed a wearable device So nothing what you do is valid if you do not translate it. So we developed a device for neuropathic pain in trigeminal neuralgia, which got funding from me. It was very pittance, but still we got it, and it's a prototype now. So it is based on a very simple principle. What do you do when you hit your leg against a, say, bed post? What do you do? What is the first reaction that happens? What do you do? Shout. And the next thing? Next thing. Suppose you hit your hand somewhere. Okay, I hit my hand here. What is the first thing that you do? You do this, right? You do this. This is the same principle. You are activating touch sensation to prevent pain. The touch fibers are faster conducting fibers. The fibers, nerve fibers, yes. Something, huh? Okay. So the touch fibers, they conduct at 70 to 120 meters per second, whereas the pain fibers conduct at just 2 meters per second. It's like putting a roadblock. If I excite the touch fibers by rubbing, so then I am stopping the movement of the pain to the brain. Until your brain, the sensation of pain reaches your brain cannot perceive pain. How nice? What is the best example for this? People in battlefield do not feel pain. Have you seen? They will fight with a lot of injuries. So till the brain does not perceive, you cannot feel pain. And that simple principle we utilized for this trigeminal neuralgia, which is a very debilitating pain. So we used vibration to block the sensation of pain. And finally, I could do something which is more patient-oriented, and that is my journey started with intraoperative neurophysiology. If you see here, this is a brain with electrodes, and now we can directly put the electrodes on the exposed brain and take the EEG from there. So it's a beautiful, marvel to see the brain and record EEG waves directly from the surface of the brain. And also during surgery, the patient is kept awake. We keep on talking to the patient, asking questions, and while the surgeon operates from above. So that is the next frontier, and which excites me a lot. Telling the surgeon, okay sir, you are near the language area. And that's how I have satisfied my clinical urge. So this is clinical neurophysiology, and it is now a big deal here, because in all neurosurgery orthopedics cases where you are touching the brain and the spinal cord, you require this monitoring to prevent neural damage. Okay, so brain tumor. Okay, I remove the tumor, patient is not able to talk. You have killed the social life. Quality of life has become more important. That is why these procedures have now taken prominence. So with this, I will give you a life lesson. The brain never lies. If you learn to listen to its signals, it will always tell you the truth. So we need to listen to our brains, and science is not by the publication count, the ResearchGate scores, or the Google Scholar citations. I feel science is through the legacy. If it can be continued, even if one student excels after you, your job is done. So my happiest moment was when my PG student got the same award which I received in neurophysiology for the best paper in neurophysiology. When she got it, it was a happier moment for me. And so I would like to end for you today, students. Your curiosity is valid. Your questions, no matter how naive, they matter. Science does not require a perfect path, just questions and the courage to stay curious. So start where you are. Use what you have and do what you can. And even if there are frustrations, stumbles, I will quote my ex-HOD, Professor Mohapatra, the sky will not fall. जिस दिन गिर जाएगा ना उस दिन देखेंगे बाकी सब problems can be handled. Rest all can be handled till the sky falls. So with that, thank you for giving me a patient hearing. Thank you so much for sitting through your hunger pangs. Thank you.
Thank you very much, madam, for such a motivational deliberation and for a very interactive session. And at this juncture where everybody was feeling sleepy and hungry, your presentation actually awakens everyone. And thank you once again for making us understand the science behind the sleep, food, and the other relevant aspects of our life. So with this, we have almost come to the end of today's celebration. But before we conclude, I would like to invite the Director, Research and Development, Professor Joydev Dinda sir, to kindly present the formal vote of thanks. Sir, please.
Good afternoon all. Very respected Honorable Vice Chancellor, Professor Yogeshwar Dandapat, in absentia Chairman PC Council, Mithali Madam, Register Madam, and distinguished speaker. Actually, Department of, I think, R&D cell has to take the opportunity of the motto of the Government of India, Women in Science, Catalyzing Viksit Bharat. Utkal University, the mother university of Odisha, take this opportunity to celebrate this National Science Day to honor the woman. Really to say, when I am the student of master, then we have only 3 girl students with us. But now I become the teacher in 16 to 18 girl student. So at least that reflect that women are coming. So we are expecting much more girl student to come this science stream to explore, to serve the nation in a better way. Anyway, so today we are very thankful to the, very enriched talk given by Professor Samir Kumar Patra, Professor Vedandas Mohanty, Professor Sushil Kumar Singh, Professor Priyadarshini Mish. And I think everybody, you delighted having this kind of knowledge, and the knowledge you today received, that you will disseminate in the future day. So we are very, very thankful to the esteemed speakers. Within their busy schedule, they came here and they gave up their valuable time. Sir, we are very thankful to, on behalf of the Utkal University, we put your respect to yours. And I am very thankful to the student, at least the student who are at least at the end in the present in the hall mostly. We are very thankful to the student for coming to this day. We are very thankful to our administration who have given the full support, and our staff of research and development cell. And lastly, if I didn't say, our deputy director, Dr. Priyanka Khurana madam, she did excellent in our cell, and she did lot of to organize this kind of seminars. And thankful to, I am very grateful to the student. At least in the hall you fill up and you give your dignity, you give every respect. So once again, thank to you and look forward in the future. Okay, thank you all. With this, we complete the session, so please. So now we are arranged for some small tiffin. Please come in a decent way to receive this one. Okay, thanks.